crowd we've had yet this spring. I attribute it to you and your topic and not to the beautiful weather.
Well,
Then the folks at home, let's see. Tom Kinsley and Sandy are longtime Wednesday night at the lab folks. Tom used to work with selling optics. Sandy's all around electrical engineer. And that's me, and that's Paul. And he was a high school teacher and school administrator, and he's watching from home. How's that, Carol? Great. All right. Now I get to ask you the 5 questions.
All right.
Jennifer, where were you born?
I was born in Carrollton, Texas. It's north of Dallas.
And then where'd you go to high school?
I went to Hebron High School in Carrollton, Texas.
All right. And there, where'd you go for undergrad and what did you study?
I went to Texas A&M. I studied biochemistry and genetics.
And then when did you come here? And or did you go anywhere between A&M and here?
No, I came straight here.
And now you are in the Department of?
I am in the microbiology doctoral training program. So it's in the College of Ag and Life Sciences, but I do my research in the School of Pharmacy.
That's what's interesting. So one of the major things that still impresses me about UW-Madison is the crosstalk between the College of Ag and Life Sciences, the College of Letters and Science, and the School of Medicine and Public Health, the School of Pharmacy, the School of Nursing, the School of Veterinary Medicine. The fact that we're all on one campus, pretty close to each other. The building that you work in is the Microbial Sciences building.
Oh, I don't get to work there. My program's based in there. It's a beautiful building, but I work in Rennebaum Hall, so
that's
the
a pretty
Pharmaceutical
nice building
Sciences
too.
building. Yeah.
So today you're going to talk with us about investigating bacterial biology with CRISPR tools and antibiotics. Please join me in welcoming Jennifer Tran.
Hi, everybody. I think we have a pretty diverse crowd. So I think for some people, maybe this is a little too basic. So feel free to interrupt me with very difficult, more in-depth questions. I do want to say, we went around the room, my research is very both biology and computational, but I'm not a computational expert. So please don't quiz me on coding because I'm not good at it. All right. So just to get started, see. All right. So if you get nothing else out of my talk today, fall asleep, something like that. The most important thing to know is that what we do in our lab is figuring out how to combine antibiotics and genetics to better understand how disease-causing bacteria functions, why antibiotics do or don't work, and then the end goal, the grant funding goal, is to identify new ways to target and kill these bacteria. I came to UW-Madison interested in infectious disease. Infectious disease can be caused by a number of things. So bacteria, so those are things like E. coli, staph, strep, viruses like COVID and the common cold, fungi, you could think athlete's foot or yeast infections, parasites like roundworms, tapeworms, and then prions like mad cow disease. I was particularly interested in bacteria. UW-Madison has an amazing bacteriology department, and that's part of the reason that drew me all the way from the South here. And just a nice graphic here for really putting bacteria into perspective. If this here is a human hair, and this is a grain of sand, a grain of salt, bacteria are all the way down here, 1 to 10 microns in length, so very, very small, can't be seen with the naked eye. And these small single-celled organisms are everywhere. They're ubiquitous in the environment. So you have good bacteria, which I think are kind of like the rage nowadays. You hear about probiotics and that kind of thing, helping your gut bacteria be healthy. So you have good bacteria, you have neutral bacteria that are kind of just living their life out in the environment. And you have the bad bacteria, the bacteria that cause infections. And the way that we historically treat bacterial infections are with broad range antibiotics. So these antibiotics, you take them, they will clear bacteria, they will kill the bacteria. But the thing is that, over the past few decades, there's been really an overuse of antibiotics. So they're used in agriculture, livestock, sometimes they're overprescribed. And in maybe the last 2 decades or so, we've seen like this huge rise in antibiotic resistance. So basically, you take an antibiotic to clear a bacterial infection, but those bacteria have seen the antibiotics already. They've evolved and adapted to no longer be affected by them. So this is a problem that our lab wants to address. Like, can we find solutions to this threat of antibiotic resistance using genetics? And so for some of you, this might be like a little refresher on biology knowledge, but the very core of biology and genetics is the central dogma. So you have your DNA, your genes. Those genes get transcribed, basically read into RNA or mRNA. That RNA is then translated into proteins, and those proteins are what go and perform cellular functions. And so really, when you think about genetics, it's like by studying this DNA, we can get an idea of what happens downstream. So like how a cell writes in its genetic code to perform important functions is very important to understand, and really a great way to kind of approach these problems, especially when it comes to bacteria-resistant antibiotics. And our lab takes a systems biology approach to dissecting genetics. So if you imagine this circle here is a gene, classic genetics research often like really focus on like a single gene or a handful of genes, and they do a deep dive. So what does this gene encode for? How does it get turned on and off? Well, what are these things that affect this one particular gene? But our lab kind of takes this bird's eye view, because we know one gene doesn't live in isolation. They're really involved in these like larger pathways where there are multiple genes working together to perform a function, say cells digesting or taking in oxygen. And the thing is, though, that like if we zoom out all the way, it's even more complex than that. So all these different pathways are interconnected to form this genetic network. So pathways talk to each other in order to keep the cell running and alive. And we're interested in this network as a whole. So in a bacterial cell, all the pathways interact in some way. And I think that really makes sense if you kind of look at what a bacterial cell looks like. So unlike eukaryotic cells, it doesn't have compartments. It's densely packed. All the pathways are working in close quarters to help the cell survive, and these single cell organisms survive on their own. And really, these pathways are really intertwined. So you can imagine this, if this yellow is DNA here, it's being transcribed into RNA, translated into proteins, all in this one little cell. And sometimes stopping one part of the network because it's so intertwined can cause cell death. And so those pathways and those genes are considered essential, hence the name. So here are some examples of essential processes. So for example, DNA replication. Can the cell make more DNA? Metabolism, is the cell eating? Can it make those proteins? Translation, like we talked about. And then the cell membrane and the cell wall, which kind of hold everything together. They surround the cell. And we actually exploit the essentiality of these processes through antibiotics. So there's a list of some common antibiotics here, and they specifically target these essential processes to kill bacteria. But the thing about antibiotics is that even though they target, one gene or one pathway, it's not just the one thing that's affected. It's also all the other genes that are connected as well. And so, the approach for my project in our lab is like, if we can study this genetic network, like we will be able to learn and better understand how current treatments affect bacterial biology as a whole, how different treatments could work together to better kill bacteria. So really trying to target the entire network. And we can find new weak points in this network that maybe could be new targets for new drugs. So our lab studies this kind of thing in multiple different bacterial pathogens, so disease-causing bacteria. But for my project, I work with Acinetobacter baumannii. I like to call it, for now, I think, the scariest bug you've never heard of. But I think that it's becoming a more common name, especially in the news. Acinetobacter baumannii, or A. baumannii, is a bacteria that's considered an urgent threat by the CDC because there have been clinical cases where antibiotics don't work. So none of the antibiotics available on the market can kill this bug. And, it causes wound infections. It was really started that way because in the 90s, it spread to military hospitals during the Iraq War. But now it's become really this hospital-acquired infection. So you'll go in for a surgery and then you get a secondary bacterial infection while you're there. A. baumannii now in the US causes ventilator-related pneumonia. That's the most common way people get these bacterial infections. And I think one thing to note here is that these numbers that they have here, like 8,500 estimated cases, 700 deaths, are actually understated. So the CDC used to release these updated numbers every year, but with COVID, they've been really behind with getting the bacterial numbers together. But because of COVID, with the uptick in ventilator use in hospitals, there's also been a corresponding uptick in Acinetobacter infections. While we're here, A. baumannii is very tricky, I think, as a bacteria. So it has some very unique biology to it that makes it resistant to all these drugs. But, even though it's a serious problem and it's a very different kind of bacteria, we know very little about its genetics. So the goal of my project is to figure out how, the essential genes and the pathways in Acinetobacter interact. Because the idea is if we can actually map out this network on the line, we can maybe find new drug targets because it's resistant to all drugs. It can be resistant to all drugs on the market. Or it could inform us maybe on better combination therapies to treat it. So what mix of antibiotics can we use to achieve a better effect? So I said I'm interested in essential genes. Traditionally, genetic approaches often involve knocking out or turning off genes to study them. So things like transposon mutagenesis or deletions. And normally you get rid of the gene. It doesn't make the RNA, it doesn't make the protein. And then you can see, well, there's less of this teardrop protein here. So maybe we can see this gene must be involved in making this teardrop protein here. But the thing about essential proteins is that if you go through that whole process, the bacteria die because that process was essential. And now we don't have bacteria to study. We just have an empty Petri dish or clear flask, cleared culture. So to look at essential genes, we use a tool called CRISPRI. So the CRISPR that you might normally hear about uses a single guide RNA. It directs this protein called Cas9 to a specific matching piece of DNA. And Cas9 normally cuts DNA. But we can't cut the DNA for the problem I just mentioned. So we use CRISPR-I, so CRISPR interference. It's similar, but it uses a dCas9 instead. It's catalytically inactive or dead. So the dead Cas9, it doesn't cut. Instead, it just sits on the DNA and blocks transcription. So it blocks that process of DNA to RNA. So RNA polymerase makes that RNA, it comes along, dCas9 is there, and it blocks it. But the thing about CRISPR-I is that sometimes, very occasionally, the RNA polymerase will knock the dCas9 off. So there's still a little bit of read-through, so the cells don't die. You get a very, very little bit going through, so that essential function that the gene product is performing can still happen at very low levels. And actually, we can genetically engineer this by changing the nucleotides in this single guide RNA. So we can design what genes we are going to target. And we have designed guides to target all of the essential genes in A. baumannii. And the way we do that is we have our CRISPR-I machinery, and we can actually just integrate it straight into the genome of A. baumannii. For genetics purposes, this is actually pretty important. It's kind of a little special feature of the tools we use in lab, because a lot of labs will not put things in the genome, and then it requires antibiotics to keep DNA around. A little bit of an aside, but it's very useful for our purposes. And the system is inducible. It means we can control when it's turned on. And we do this all in this pooled fashion. So actually we have tons of guides all in this little tube, and we can go through this process and end up with a essential gene knockdown library. So we have this library where there's a bunch of bacteria, a bunch of baumannii. The strains are all mixed together, but the strains in there each have a unique guide that's going to knock down a different essential gene when the system's turned on. So now we've done like this genetic side of chemical genomics, and now we like move to the chemical side of chemical genomics by adding in antibiotics. So we have our library. It targets 406 essential genes in a baumannii, and we can turn on that CRISPRI system and then expose the library to a variety of different drugs or different antibiotics at sub-inhibitory concentrations. So we don't want to wipe out our library. We just want to see how our library reacts to a little bit of the antibiotic. And then we're interested in how the composition of the library changes. So, which gene knockdowns are going to grow better, which knockdowns are going to grow worse or maybe drop out of the library entirely. That'll tell us how different genes respond to different antibiotics. And, unlike my nice picture where all the bacteria are different colors, it's not like that in real life. So we need a way to measure the library changes. And in order to do that, we use high throughput sequencing. So we can actually just extract the DNA and amplify the guide region. So I told you that we designed the guides to match different targets, but that also means the guides are unique. And so we can treat them kind of as DNA barcodes. And so by sequencing the DNA, we can then count all the different barcodes, and it tells us how many of the bacterial strains are there at the beginning and how many are there at the end. And so then we can measure those changes. But what we get back are a string of sequences that we can count. But the question is how do we make sense of the biology? And so I'm gonna start off with kind of a snapshot of what the phenotypes we see in our data. So say we were looking at just one gene knockdown in one antibiotic, and I'll call it drug A here. And if we see that it's dropped out or has fewer strains after the antibiotic treatment, then we say this gene knockdown is sensitive to this drug. So it's represented here by a red square. And if in a different antibiotic, if we call it drug B, we see that same gene knockdown grows better, the knockdown is more resistant. So it's represented by blue. And we can use these kind of colors or a heat map to look at gene knockdown in all of the antibiotic conditions. So each square here would be a different antibiotic gene phenotype. And so from here, this is just a little cartoon. We can zoom out. And this is just another little piece of my actual data. So we have multiple gene knockdowns here over multiple different antibiotics. And again, red is sensitive, blue is resistant. And for these heat maps, these things are clustered. So it means we've mathematically grouped together gene knockdowns that look similar across all the drugs. And we've grouped together on the axes drugs that look similar across all of the knockdowns. So from here, we can start to identify patterns that might tell us more about the biology. So one example here, these antibiotics here are cell wall targeting antibiotics. These are things like beta-lactams, like penicillin. And they look similar. They have a similar pattern across this axis here. And actually here in this red, what's really sensitive are the cell wall knockdowns. So the target of these drugs, when you knock them down, the bacteria become more sensitive, and that makes sense. But patterns like these can also point out how drugs that we don't know how they work, like drugs of unknown mechanism, based in our screen. So we have a couple of drugs, we don't know what they do. If we can see if they look like drugs we do know, then we might be able to figure out how they work. If you look on the other axis, genes with similar patterns across all the antibiotics also have similar function. So the genes here are actually all NADH dehydrogenase genes. It's involved in oxidative phosphorylation, which is not super important here, but I have, if you want to talk about oxidative phosphorylation later, feel free to come to me. But these genes cluster very tightly together. They have strong phenotypes across antibiotics of varying mechanisms here. And actually, like, we think that this pathway is important for how antibiotics get into the cell, because it's involved also in how strong the membrane around the cell is. But one thing I want to note is that like strong phenotypes like this could point at like something being a good drug target. So NADH dehydrogenase could potentially be a good drug target for baumannii. And then, like I mentioned with the antibiotics, we can also see genes of unknown function. If they cluster well, if they look like genes we know what they do, then we can start to figure out what the unknown genes do. So, what if we look at all my data? It really starts to kind of get difficult to see what's going on here. Again, things are like clustered, but really, unless you like print this out as a tapestry and hang it in your lab and have your PI go over with like a little magnifying glass, it's going to be hard to tell anything. I will zoom in here and just show that, we see these clusters that are really positive. They indicate that this experiment worked. So here we have a cluster of a lot of genes that are involved in their ribosomal proteins. So basically they're involved in that process of translating RNA into protein. And it's great that they cluster all together. We know what these genes do. So it's a proof of concept here. And usually here is where my committee will get really confused and be like, why did you use so many antibiotics? It seems like that was too much of your time. So I'm gonna quickly address that here. Imagine if we, instead of 50 conditions, we only had 2. And I think you can see here, it would be really hard to say that all of these genes have the same pattern with just these 2 conditions. And the patterns really don't jump out. But when you add in all the chemicals, you can see over here, these are all blue, over here, these are all red. There's kind of a unaffected section here in the middle. And it's the patterns across all the conditions that's really important. So the question is, my PI still wants to print out the tapestry, but what if we don't want to do that? And what if there's a better way to visualize this that's not just a bunch of little colored squares? And we can use the patterns that I mentioned to make a better visual for how these genes interact. So if we correlate the patterns of the genes, so basically how similar are those gene patterns? We can input this data into this computer program, and it generates this visual where each circle here is going to be a gene. It's called a node. And these nodes are going to be connected by edges. And the edges are basically that numerical value I told you about how similar the gene patterns are. So these connections are drawn between genes that are highly correlated. So for example, these genes here in this little triangle would be genes that are highly correlated with each other. They look very similar across all the antibiotics. And then genes that are a little farther apart, but are still connected, they're still related, but maybe not as highly correlated. And so I guess one thing I'll note here for the computer programmers in this crowd is that the number of connections and the shape of the network changes a lot based on the thresholds that you set. So keep in mind that this is just one way that I can look at the data, and there's probably thousands of other ways that I can visualize this that depends on what kind of numbers I'm putting in. So in my case, I'm looking at the top 20% of all the connections here, and I can generate this network. And again, it's still a lot of data, so it's kind of hard to see. If we zoom in, here's those same genes I pointed out earlier, the ribosomal proteins, and they're all part of this genetic network. But we can find some interesting things, and we did. And so from here, I'm gonna share some short stories of some interesting things that we've learned. My lab likes to call this hypothesis generation. So we're pulling these giant data sets, and then from the data sets, we're gonna look at them and generate little hypotheses that we can do follow-up experiments on. So Some of the things that I'm working on, I come from a cell division background, so how cells grow and make daughter cells. So my PI let me follow up on this. But basically, this cluster here are some genes that we know are involved in how cells divide. But we also see some things that are a little bit different that we don't know. So there are some genes here that are Acinetobacter specific. There's very limited experimental evidence. We're not really sure what they do. And then there's also this uncharacterized gene that's predicted to make a protein that might bind to the cell wall. So when cells divide, it's very important that they make enough of the outside cell membrane, otherwise they'll explode. And just looking at these genes, like how can we figure out if they actually are involved in cell division? One way is by pulling those knockdowns out of the library and looking at them individually. And we can see if they show similar phenotypes to cells where we know division is inhibited. So here's an example of like normal division, where your chromosome is going to get replicated. It will get sequestered into 2 forming compartments. And then cell division proteins help form this little constriction ring or septum, and it leads to 2 daughter cells. But if you inhibit the cell's ability to start the ring or to finish division, what you end up seeing are these really like long cells. So these are cells that can't divide. It's a very common phenotype in the cell division field for bacteria. And so here's some microscopy to show you what that looks like. So here's Acinetobacter. It's just a control. This is what they look like normally. And then when you knock down something that we know is involved in cell division, they get that really kind of long, elongated phenotype. And if we look at our unknown genes, we can see here that they definitely appear elongated, but maybe not to the extent of this known division gene. But we do know, now we have proof that they are involved phenotypically in cell division. And we're currently doing more work to characterize this, and I'm happy to talk about, but it's maybe like a little, a little bit jargony, I'll say, yeah. So back to the network. Here's another little story that we're also thinking about. So this was an unknown gene. All the genes in like bacteria are often named this awful combination of like letters and numbers if they can't figure out what it does based on like just looking at the sequence. So here was an unknown gene, and in our network, it's connected to these genes that are involved in DNA replication. So making more DNA so the cells can divide. And like, we're like, well, it's unknown, but it's connected to all these division genes, like, or these polymerase genes, these DNA replication genes. It's probably involved in DNA replication. And we did structure prediction, and sure enough, it like the structure prediction says like, hey, it does look like something that's involved in DNA replication. But the thing is, for the field of like kind of this computational biology, when people are looking at bacterial DNA sequences, they're saying like, well, based on ACs, Ts, and Gs, or like based on just the sequence, we're going to try to predict what these genes do. And sometimes that prediction is bad. And I think this is just one way to show like this, doing these kinds of experiments in your own special little bug can improve that kind of automatic annotation that happens in the field. Okay, so one last little thing I think that I'll leave everyone with is that these are looking at the connections between all of the genes based on the patterns across all of the antibiotics I used. But we can do it the other way around. What if we connect antibiotics based on how the gene knockdowns look across all of them? So these are correlations between chemical effects on gene knockdowns. So this is kind of a chemical network, if you want to call it that. Not all of the chemicals are here. It's a threshold issue for creating these networks. So if anyone has any ideas on that, that would be great. But I think that the cool thing about this is that it can point to mode of action. So how antibiotics work. So like over here, these are all penicillin-like antibiotics. It's great that they cluster together. There is one weird one that is not involved. It's not penicillin-like at all. And we think that it has something to do with how well antibiotics can get into the cell. So we think that there's a permeability story here, like bioavailability. Can the drugs get to their target to work? And that's something that we're currently doing experiments on. But I think a really cool thing here that came out for me is carvacrol and thymol. These are chemicals that have been purified from herbs. So we don't really know what they do. There's been some literature that suggests that they are antibacterial, and we do see that Acinetobacter can be killed with enough of these chemicals. But by looking at how they interact or how they connect to known chemicals, maybe we can figure out what they're doing. So I think that's pretty cool. Ciprofloxacin and EDTA here are potentially, we don't really know the story, but we're doing follow-up experiments to figure that out. Okay, so just to kind of summarize everything I talked about, and there are more stories if you want to ask, but there's a lot. But essential genes are the key to antibiotics and how they work. Chemical genomics combines CRISPR tools and genetics with antibiotics to analyze this essential gene network. And chemical genomics in baumannii, which is a serious disease causing bacterial species, revealed some new biological and new antibiotic findings that we're currently working on. And I'll acknowledge my PI, Jason Peters. Amy Banta is the research scientist and his partner in the lab. And she does like she has all the technical expertise. And Ryan is like the IT computational guru. He's actually an economist. So he comes to us from a very different statistical analysis background, which is really great. And he's been a great resource. And then here's the picture of my lab and my funding. And then we can open it up for questions.
Question.
Yeah, I had one. A question and a comment. First of all, when I see network stuff popping up all over the place in the last 10 years or so, whether it's economics, sociology, you make the networks, and then you can use just theoretical things about mathematical representations of networks to apply to all these different fields. So I find that really interesting to see where it pops up.
Yeah.
And the question I wanted to ask was, with this baumannii, did people try to come up with some kind of bacterial phage, the virus? Oh
yeah, absolutely. So my undergrad research was in bacterial phages. So I don't know if you've heard or read the book A Perfect Predator, but when I was in my undergraduate, I was in a lab at Texas A&M. We studied phages, and there was actually an FDA emergency authorization for Stephanie Strathsty's husband, Tom, and they were professors at UC San Diego, I believe. He had gotten an baumannii infection. It was multi-drug resistant. They couldn't treat it. He was dying. So they got an FDA emergency authorization. Our lab helped purify some phages against the strain of baumannii that he was infected with, and he was saved. He was cured. So I think it was a really cool story, and it was really cool to be there while that was happening. And phage therapy is this thing I think that they're working on getting through FDA approval to try to be more commonly used for these infections that can't be treated. So
do you work on that at UW at all?
Do I work? I don't work on it. There are other groups that work on phages, but I think they take a little bit of a different approach. Yeah. So I came from a phage basic biology background, and then it just happenstance is now very useful. Now that antibiotics are kind of becoming less effective. But here they take more of an engineering approach. So they're if we have phages, can we engineer them to be able to treat different bacteria? And that way we can have a repository of phages in the future. Yeah, it's a little bit of a different approach because we took let's find it in biology because we know it exists out there somewhere approach at Texas A&M.
Can I ask real quick, how do they get the phages inside the person?
They just injected it directly into, so he had massive organ failure, and there were spots of growth of A. baumannii on certain organs. They were just able to actually just flush it directly into the spots of infection. I think they also did IV. So it was kind of like, I think, a wild ride because no one knew if it was going to work.
And does that make him, his immune system next time respond to those phages?
It's hard to say. I believe that probably most of those phages would eventually get cleared from his body. And also based on like the paper that came out of that, it does, like, the phages were very important to bringing down the bacterial infection, but then his immune system took over to really finish the job.
Yeah.
So yeah, so I think that he's very immunocompromised now, because I think that a lot of his organs didn't quite fully recover because he was in such a bad state. But I imagine that he probably has some natural immunity now to the Acinetobacter strain, at least that infected him.
I'm gonna Google the guy's name. What was his name?
Stephanie Strathey is his wife. I'm like in close contact with her, but her husband's Tom, but his last name's not Strathey. Yeah, but the book is a perfect predator. It's like she wrote a novel on it, and so I think that's pretty cool. They have this whole we're kind of going off this tangent because I love phages. They have this whole center now that their entire thing is getting FDA emergency authorization for people with life-threatening bacterial infections, and then finding those phages, getting them purified, and sending them out. So I think it's a pretty neat story. And it all started with A. baumannii. It's why I wanted to study it when I came here. Yeah.
Have you read the book Aerosmith?
I have not.
You know the book?
No.
Sinclair Lewis, 1926. He was a young man who wanted to go to medical school. He did work with some biologists, and biologist was looking at whether, or entertaining whether bacteriophages could cure disease.
Yeah.
And he decided to go into research.
Yeah, that's really cool.
So that was 1926.
Yeah.
So it's a classic.
I should read it.
It's a great novel.
It's a great novel. Yeah.
He's the same author that did like Main Street,
Yes, yeah.
Babbitt, Elmer Gantry.
Yeah.
So his books are all great, except they always have a love interest in them.
Yeah. I think phage therapy would have stayed around if it weren't for the discovery of penicillin. The reason that like antibiotics overtook phage therapy, which it was kind of like burgeoning before World War II as kind of a potential option. But they didn't really know the biology back then. So phages are host specific, which means they only target some bacteria. So when you had like a cocktail of phages that someone had, prepared, and you have it in a little vial and you take it, sometimes it doesn't work because the bacteria that you have isn't one that the phages can infect and kill. So when it comes to like antibiotics, which are broad spectrum, they will kill all the bugs. Then, those seemed like a better option at the time. Yeah.
It's my understanding that the country, Georgia,
Yes.
does have a repository.
Yes.
Phage, and they do use phage therapy regularly.
Yeah.
And I have 3 related questions for you.
Yeah.
What's your next step in your research? How soon before you expect to finish your PhD?
Oof.
And what's your plan to do next?
The big questions. If we're thinking small scale, I would really like to finish up this cell division project and then publish that. We also have another project in the lab on oxidative phosphorylation, which I didn't really talk too much about today, but I'm working on some cool assays with that. I'm looking to finish in spring of 2024 is the goal. My PI says it's a little ambitious, but I think that's okay. I think it's good to be ambitious. And next steps, I'm actually like kind of pivoting. So I really enjoy like science communication, and I like speaking to people, but I don't think that the pipetting is really for me for the rest of my life. I have weak wrists, you know. So I'm going to be moving into consulting. So ideally like life sciences consulting, potentially branching out from there. So
Thank you.
yeah.
Have you thought about the AAAS Congressional Science Fellowship?
I did think about it for a while, but I think I'm more interested in like speaking to scientists and startups and maybe not politicians. Although I have done like a little bit of like science outreach with like UW-Madison, like Day at the Capitol, but I think it's a tough climate sometimes. So yeah.
Other questions? Have online.
I don't think there are any questions in the chat.
So what's it look like when you grow all these different strains of bacteria with all these different types of antibiotics? How do you do that?
So it looks less cool than the heat map I showed you. It all happens like in either flasks or tubes, and then they're just growing. The library is growing in this little bit of liquid. You add a little bit of clear liquid to that, which is the antibiotic, and then you continue to grow it. And then I just collect what was in the tube. Like I collect a little bit in the tube before I add the antibiotic. I collect a little bit of what is in the tube afterwards, and then we send that out for sequencing. And I just do that in parallel for all the different antibiotics. So say we have like 50, 100 tubes if you count all of my controls. Yeah, well, sometimes I spread it out over multiple days. It's a lot of pipetting. But
so you do this, and it's this little Erlenmeyer flask with that you swirl around, and
yeah,
the little ridges in them that aerate it.
Yes and no. So actually someone else in my lab has been working on growing Acinetobacter to like larger concentrations for other experiments. And so it's actually, I didn't say this, but it's an obligate aerobe. That's why it does so well in the lungs for like lung infections, because it needs oxygen to survive. It can't survive when there's not oxygen. So normally you would think the little flasks with the little ridges in them, those are supposed to help aerate. But weirdly enough, for this bacteria, it gets really foamy in the aerated thing, and the foam kind of creates this layer to where the bacteria sometimes doesn't grow as well because the foam is preventing oxygen from getting into the flask at the bottom. So sometimes, like for larger cultures, we just use the flask without the ridges. Yeah.
So you have about 50 of those going on a shaker table at once?
Yeah, we have a big old shaker. Yeah.
Okay, so it's not in 8 by 12.
No. So actually, there are other people in my lab who do this, not with disease-causing bacteria, but with bioenergy bacteria. Look, instead of antibiotics, they use like ethanol and other things that are created and are toxic in bioenergy production, like biofuel production. And they do the plates. For my purposes, the plates don't have enough oxygen content when we're growing them to really be effective. So, yeah.
And then is this what bioprotection level, biosafety level is?
This is a BSL-2. We actually have a strain in the lab so I use a lab strain, they like to call it. So it's only resistant to, I think, a handful of antibiotics and not all of them. So the strain that I work with is not multi-drug resistant. We do have one in the lab that is multi-drug resistant, but biosafety won't let us use it. So.
Is there a reason you would want to use it?
I think, especially like in the clinical field, there are people who are using this to study genetics, and often like reviewers and clinicians feel that that's more accurate to like what you would actually see in a clinical environment, because a lot of the Acinetobacter in the clinical environment is multi-drug resistant, and there are differences between the multi-drug resistant and the lab strains. So
if you come down, have there been any of these at Madison?
yeah, there have been some. I think Pseudomonas and staph are bigger issues at UW Health. But there is one of the PIs on my committee. He is a PharmD, and he is like a consulting pharmacist for the infectious disease department at UW Health. So he has seen these at the hospital across the street.
What do you have to do if you're in the hospital and you get one of those healthcare providers?
So I think in the sciences, we talk a lot about like personalized medicine, because it seems like the way that you really want to move, because you want to treat the disease, but not like have all of these side effects. But I think it's a little bit different clinically, because when someone is sick, unlike science where you're like, I'm going to streak this out and I'm going to like see the bacteria, we're going to send it out for sequencing, figure out what it is. There's just not that kind of time or those kind of resources. So usually when you come in with a bacterial infection, I believe they just treat with, they have like standards for what antibiotics to treat with, and they usually go down the list. It usually doesn't get to the point where like we need to figure out exactly what bacteria this is and like sequence the genome and figure out like how else we can treat it until the disease is like much further progressed. So it's kind of crazy.
Rapamycin. I quite often don't say it correctly. It was an antifungal drug for it's great for jungle run. Vietnam. Turns out there's a dog study across the country as the anti-aging drug.
Rapamycin, right? Yeah. I have no, it's RAPAMYCIN, yeah, rapamycin. I've heard about it. I'm not very familiar with it. So antifungals are kind of a different beast than antibiotics. So actually fungi are more similar to humans than they are to bacteria. So the way that you treat them is a totally different beast, because you have to be, antifungals are actually more difficult because they're so similar to human cells. You really have to be careful when you're developing antifungals that you're not accidentally also killing off human cells that look more similar to the fungi. So I'm far more familiar with antibiotics than I am with antifungals. Yeah.
Other question. Well, that's a great talk. And it all working.
Thanks to everyone on Zoom.